Hello, everyone. My name is Atenea Reyes, and today I will be presenting an evidence based analysis of functional food that has gained massive popularity kefir. I guess everyone has heard of kefir by now, as it has become a staple in wellness blogs and supermarket aisles alike. However, today we are going to dive into the hard science behind it to see if it actually lives up to the hype. In this presentation, our independent variable is the oral intake of kefir, and our dependent variable is the modulation of inflammatory markers and gut microbial composition. To truly understand if this superfood lives up to its reputation, we will look at it through three different lenses: an animal model to see the biological mechanisms, an observational study to see how it affects real people in their daily lives, and finally, a clinical intervention to prove its medical efficacy. We aren't just looking for good food, we are looking for a reliable, evidence based nutritional tool. So, why are we talking about this? Why is the research relevant? Currently, we are seeing a global increase in the prevalence of gastrointestinal and metabolic disorders. As future nutritionists, we must verify if the probiotic benefits observed in laboratory settings actually translate into clinical improvements for human patients. By comparing these three levels of evidence, animal models, observational data, and clinical interventions, we can determine the biological consistency of kefir as a functional food. As a functional food, this comparative approach allow us to see if the anti-inflammatory mechanisms we observe in the lab hold up when subjected to the complexities of human lifestyle and disease. Kefir is particularly interesting in this context because it's not just a simple probiotic supplement, it's a unique, complex symbiotic matrix of bacteria and yeasts. This living food matrix may offer superior survival rates for beneficial microbes as they pass through the digestive tract, potentially making it more effective than isolated supplements at modulating the gut immune axis and restoring the intestinal homeostasis. Now, let's dive into our first piece of evidence, which is an animal study. This is where we see biological heavy lifting of research. We begin with a study by Van de Woo and their colleagues, published in 2020 in the journal Microbiota. The aim was that this research, the aim was to investigate the specific impact of kefir on gut microbiota composition, systematic immunity, and even host behavior using healthy mouse models. To achieve this, the researchers used male C57BL6J mice, which were supplemented with either two distinct types of kefir, referred to as kefir A and kefir B, or plain milk as control group for a duration of 3 weeks. To analyze the internal changes, they utilized 16S RNA sequencing for a detailed fecal microbiota analysis and measured systematic cytokine production directly from the mesenteric lymph nodes. This methodology allows us to see the deep mechanistic why behind kefir's impact on the immune system before we translate it to human data. Now, let's analyze the technical results in Graph A, Alpha Diversity Plot. We observe the Shannon Index, which measures species richness and evenness. Note how the ka'akum, kefir, significantly increases diversity compared to the milk alone with a p-value of 0.014. Now, in graph B, Bray-Curtis MDS plot, we see the beta diversity. These points represents the distance between bacterial communities. Notice how kefir groups, the orange and the blue, cluster differently from milk group, proving that kefir truly reconfigures the intestinal ecosystem. Finally, in graph C, we identify which specific bacteria change. Kefir consumption significantly increased the abundance of Akkermansia monsiphilia. This is a superstar bacterium in nutrition due to its anti-inflammatory properties and its vital role in maintaining gut barrier integrity. This finding provides the mechanical basis of how kefir protects against tissue damage. This way, pathological bacteria can't enter the gut barrier. Moving from the controlled environment of the lab to the complexities of the real world, we examine an observational study. While animal models are excellent for revealing biological mechanisms, observational studies are essential for understanding how dietary habits function in a free living human population. For this level of evidence, we look at the research conducted by Gonzalez and her team. This cross sectional observational study involving healthy adults, it's important to note that this phase we did not intervene or provide any specific treatment. Instead, we simply observed the participants' existing eating patterns and analyzed how those natural choices relate to their overall health biomarkers. This study deliberately moved away from experimental approach of giving people kefir. Instead, it focused on observing 150 healthy adults to determine if their natural habit intake of fermented dairy products was already providing a protective effect. To capture this data, researchers utilized food frequency questionnaires to meticulously track what these individuals have been consuming over a long period. Following the dietary assessment, they collected both blood and stool samples to measure critical inflammatory markers such as CYP-reactive protein and various cytokines, providing a comprehensive view of participants' systematic inflammatory state. In graph A, we see that fermented foods represent 33% of total dairy intake in population, with yogurt being the leader at 75%. Graph B shows us that the whole natural yogurt in predominant choice, 57%. The interesting part happens when we correlate this with inflammation. This heat map is key. It shows us the relationship between dairy intake and other food groups. Look at the red squares with asterisks. The consumption of fermented dairy, including kefir, correlates with lower levels of C-reactive proteins and fecal calprotectin. This means that in the real world, individuals who consume these foods habitually maintain a much lower baseline inflammatory state. What this tells us about people who already have an habit of consuming kefir and yogurt, they have a significantly higher Shannon index and lower baseline inflammation. This suggests that kefir isn't just a quick fix, it's a long-term insurance policy for your immune system. Finally, we reach the gold standard of clinical evidence, a randomized controlled trial. This study by Belixi Koyu and colleagues, the aim of this research was to evaluate whether daily kefir consumption could effectively improve gut microbiota composition and reduce systematic inflammation in a specific clinical population, patients with metabolic syndrome. This is a critical group to study because metabolic syndrome is a condition fundamentally characterized by chronic low-grade inflammation and metabolic dysbiosis. The researchers essentially wanted to see if kefir could act as a therapeutic tool for people who were already in a heightened pro-inflammatory state. Regarding to the methods, the study involved in 62 patients who were randomly divided into two distinct groups. The intervention group drank 180 milliliters of kefir daily for 12 weeks, while the control group consumed the equivalent with unfermented milk. This was a comprehensive evaluation. The researchers measured everything, including anthropometric data like VMI, clinical markers like blood pressure, and biochemical markers such as pro inflammatory cytokines. Let's look at the relative abundance graph. Compare the before and after bars for the Kefir group. You will notice a significant increase in the Phyllium actinobacteria, the purple section. Furthermore, there was a favorable modulation of firmicutes and bacteriates ratio. In clinical nutrition, this translates into a healthy metabolic profile, less prone to obesity and diabetes. The PCOA axis diagrams A and B, we observe how the samples from patients who consume kefir shifts and separate from the unfermented milk group. This confirms that kefir acts as a safe nutritional intervention capable of reversing chronic inflammatory markers such as TNF-A and IL-6 in just 3 months. To wrap everything up, let's compare what we have learned across these three distinct levels of scientific evidence. When comparing the three studies, we found that kefir consistently modulates the immune response toward a healthy anti inflammatory state. In contrast, each study informs our nutritional practice in a unique way. The animal study gave us a deep cellular mechanism detail, such as specific lymph node responses what would be impossible to measure in humans. The observational study demonstrated that the power of the long term habits, showing us how diversity in microbial forest is built through years of consistent consumption. The interventional study provided us necessary clinical proof demonstrating that even in a 12-week target program, we can effectively reverse inflammatory markers in sick patients. To finalize, let's revisit our initial thought. I guess everyone has heard of kefir, but now we have seen the evidence that supports the fame. From the rats and the animal study, we learned about the physical protection and reduction of oxidative stress markers like MDA. From the population study, the observational study, we saw that the long term consumption is key to the alpha diversity and resilient microbiome. From the patients, the clinical trial, we confirmed that 180 milliliters a day can reduce the severity score of metabolic syndrome and lower pro inflammatory cytokines in just 3 months. The evidence is consistent across all biological levels. Kefir is a safe, effective, and evidence based tool for nutritionist toolkit. It bridges the gap between traditional wisdom and modern clinical evidence. By understanding the variables we analyze today, intake as our cause, and inflammation as our effect, we can provide better science-based care of our patients. Um, this research demonstrates that we are not merely recommending a trend, but a validated clinical intervention that targets the gut immune axis at a mechanistic level. As future nutritionists, It is our fundamental responsibility to translate this complex biological science into practical, actionable response. We must ensure that our recommendations are grounded in evidence. Thank you so much for your attention. This was very very interesting to learn. These are my references, and yeah, that's it.